Hello, my dear students. I am ready to begin exploring with you the concepts of mitosis and meiosis. Those are two different forms of cellular division. One of the things we need to understand, though, before we can look at mitosis is the relationship between structures that appear inside of a cell when it's dividing, and those are going to be chromosomes and chromatids. Chromosomes are not visible when the cell is not dividing because they're going to be in an expanded state called chromatin. And so here in this illustration, you can begin to see the organization of a chromosome. Chromosomes are going to be, each chromosome is one molecule of DNA, but they're usually very long. I mean, these can be like several feet in length in some organisms. And so how do you get something so long to fit inside of something so tiny like the nucleus of a cell, which is going to be invisible to us? Well, the answer is organize it by coiling it up, and something that helps the cell organize its DNA is going to be these very small proteins called histones. They usually form clusters of 8 histone proteins, and when those proteins are clustered, then DNA can wrap around the proteins like thread wraps around a spool. The combination of DNA and histone proteins is what we call chromatin. So here's one important concept that I want you to remember. It's one of those things that not only you should know because it may appear on an exam, but it's because it's basic, useful information every biology student should know. Now, in most cells, we know that chromosomes are going to be housed inside of a porous double membrane called the nuclear envelope. But we also have prokaryotics like bacteria. They don't have a nucleus, but they do have DNA that also needs to be organized in one fashion or another so that it can fit inside of those tiny cells. In this illustration, I can show you a little bit better how the DNA in a chromosome is going to be organized. What I want to use is my little magnifying glass. So here we see the relationship between DNA and chromosomes. Here's our familiar double helix and how there are going to be 2 strands of DNA that are going to be running in a fashion that is described sometimes as a twisted rope ladder or as a spiral staircase. Notice how we have these histone proteins. I told you that there's usually going to be a cluster of 8 of these proteins. When those 8 proteins are assembled together, now DNA can wrap around them, forming these objects we call nucleosome. So here you see the histones working like a spool. The DNA is the thread. Both of those parts together now form these objects you can see with the help of a transmission electron microscope. That is a photo you see over here in this part of this illustration. Now, these nucleosomes are going to continue further coiling and folding when cells begin to divide. Now we are entering a level of chromosome organization that happens in the prophase of mitosis or meiosis. Notice how nucleosomes continue to coil and fold. There's going to be even a scaffolding that can help the genetic material of a cell get organized from 30 nanometer fibers all the way into 300 nanometer fibers. If you're wondering about the unit nano, nano means billionth. So now we're going from micrometers, which is the unit for measuring sizes of cells, millionths of a meter, to billionths of a meter. And so eventually, all this chromatin, highly coiled, highly looped, and scaffolded, is going to form those visible structures we recognize as chromosomes. Here, the chromosome you see in this illustration is how chromosomes appear when cells are in the middle of dividing. If you look at this illustration here, you can see that this is what a chromosome will look like at metaphase. And chromosomes here are going to consist of 2 identical structures called sister chromatids. Here I'm moving my hand cursor over one sister chromatid, and here's going to be the other sister chromatid. The 2 are said to be identical because one happens to be precisely the exact identical copy of the other. Chromosomes are going to be duplicated before cells divide Because when cells divide, what the cell wants to do is ensure that all the genetic information gets packaged into two cells, into two groups, and that each cell receives a complete and faithful set of instructions needed for that cell to function. Now we see a little more simplified version of chromosomes. One detail that I want to mention about chromosomes is that those units of heredity we call genes are going to be housed or located within chromosomes. So in this illustration, you can see how a chromosome can have many different genes located inside. Chromosomes can have sometimes hundreds and perhaps even thousands of genes in one of these molecules of DNA. At the middle of the chromosome or somewhere along, you're going to find a structure called the centromere. Centromeres are important for holding two sister chromatids together. So the chromosome you see here in this illustration is what we call an unduplicated chromosome. This is what the chromosomes of a cell will look like when the cell is not dividing. So when a cell finishes the process of division and begins to do all of its work, this is what chromosomes will look like. But sometime during the interphase stage of a cell, interphase is going to be the period of a cell that lasts longest in the cell cycle. It is going to be characterized also by a lot of cellular activity, a lot of metabolic activity. One of those sub-stages of interphase is going to be known as S, the synthesis of DNA. And watch what happens in this synthesis of DNA. Each chromosome, which has a shape like what you see here in this illustration, is going to make an exact replica, a copy of itself. And now the chromosome is going to consist of two identical parts joined at the centromere. And these two sister chromatids, which is what we call the objects, when they are together, are going to be counted as one chromosome. So here you have to pay attention to avoid confusion. This illustration here at the top of this frame is going to be a chromosome, one chromosome, but also this is a chromosome. So what is the difference? Big difference, right? Remember that we call this an unduplicated chromosome. This one down here is going to be named, as you see on this label, a duplicated chromosome. This unduplicated chromosome consists of only one chromatid. A duplicated chromosome consists of two identical sister chromatids. So when cells are dividing at the beginning of cell division, chromosomes will look like this. But before the cell finishes dividing, those two sister chromatids are going to separate and they're going to be packaged into separate cells. Here, you see two sister chromatids that have been separated, and when they are separated, they become independent chromosomes once again. So to avoid confusion and any kind of frustration, remember that chromosomes come in two versions. One version is going to be the unduplicated chromosome, which is what we see in cells early in interphase when they are doing their jobs. And the other form of a chromosome is going to be the duplicated chromosome, which is going to be when two sister chromatids are joined and one chromatid is the exact copy of the other one. We made copies to ensure that when cells divide, each cell receives a complete set of genetic instructions. Another detail about chromosomes is that they come in pairs. Our cells, for example, have two sets of chromosomes. One set of human chromosomes is a total of 23. In our cells, we have 46 chromosomes. That is because we have inherited one set of 23 chromosomes from our father and another set of 23 chromosomes from our mother. Chromosomes are going to have like a similar version, a paternal and a maternal chromosome. There's going to be one that looks the same. Those chromosomes that look the same in terms of size, shape, and the type of genetic loci. Those are the locations of genes in a chromosome. So check it out again. The chromosomes that have the same size, shape, and they have the same type of genetic information are going to be known as homologous chromosomes. One way we can see these homologous chromosomes is by examining karyotype. Now I'm going to use maybe my pen function to label this. This illustration you see here is called a karyotype. A karyotype is like a photographic inventory of all of the chromosomes in the cells of a person. If you can see this illustration, this is going to correspond to a male individual. You can see that right here. This is going to be the human male bands, and that is further confirmed because we can see that there is an X and a Y chromosome down here at the bottom. So in this karyotype, you can see that humans have 2 chromosomes, number 1, 2 chromosomes, number 2, number 3, to number 4, all the way down to pair 22. These chromosomes, number 1 through 22, are going to be known as autosomes. Autosomes are going to carry genes for the expression, for the development of all of our body parts and how our bodies function, with the exception of sexual traits. Sexual traits are going to be housed in a set of chromosomes known as the sex chromosomes. Each person normally has 2 sex chromosomes. Humans are going to have X and Y chromosomes. Males have 1 X, 1 Y, and females are going to have normally 2 X chromosomes. And remember that these sex chromosomes are going to have the kinds of genes that determine a lot of our sexuality, the development of our secondary and primary sexual characteristics. By primary, I mean ovaries in females, testes in males, and all the secondary characteristics that are typical of a male or a female. Those genes are going to be housed in the sex chromosomes. Now, let's look at mitosis and meiosis in the context of what they do. Now, in this illustration, what we see is the human life cycle. Let's begin with the moment of fertilization. This is going to be when a sperm cell joins an egg. When that moment happens, the union of those 2 gametes, sex cells, produces a single fertilized egg. This is 1 cell, and it is going to be diploid. Diploid is a term that applies to ploidy, the number of sets of chromosomes in a cell. So the reason this cell is diploid is because it has 2 sets of chromosomes. The zygote has received 1 set of chromosomes from the sperm and 1 from the egg. 1 plus 1 is 2. Simple math here. I love the math in biology, by the way, in case you haven't noticed. There's no trick here. So this cell, which is called diploid, is going to now undergo mitosis. Mitosis is responsible for the making of body cells. Body cells are also known as somatic cells. And from this single cell, we go through all of the stages of embryonic development, fetal development, until we become the adults we now are. As adults, our bodies now have a new function. In our reproductive glands, the testes of males and ovaries of females, a type of division known as meiosis is going to be taking place. Meiosis is responsible for the making of sex cells. Sex cells are also known as gametes, and in most living things are going to be known as eggs or sperm. Sex cells are different with regards to their ploidy. Sex cells are going to be haploid. Ha, the first part of this word here, is a short for half. The ploidy, or number of sets of chromosomes in a sperm cell, is only half of what you see in our body cells, our somatic cells. So that's why we applied that name to these kinds of cells. Haploid, they have half the ploidy of a normal body cell, which has 2 sets of chromosomes. Body cells have 2 sets. Gametes, sex cells, only have 1 set of chromosomes. Remember, half the ploidy. So what we want to do with this information about chromosomes and the role of mitosis and meiosis in the cell cycle of humans is now prepared to study the details of mitosis and meiosis. And there's a couple of videos I will provide to you so that you can understand really well how these forms of cell division contribute to precisely accomplishing all of the stages in our life cycles. There will be more to come. I'll see you next time.